This is an action potential graph, and it first starts at the resting potential where it's negative 70 millivolts, and inside is negative, and then the outside is positive charge. And the second step, it's the stimulus reaches the threshold, and the third step, depolarization, is when Na plus goes in and the K plus channels closed. And then the fourth step, it's the Na plus channels are closed and the K plus channels open to at 40 millivolts. And then 5, depolarization is when we set the charge gradient, and 6, undershoot and hyperpolarization is when the K plus channel closes slowly. And then 7 is when it resets and Na plus and K plus pump with ATP.
The action potential is generated as Na plus flows inward across the membrane at one location. The depolarization of the action potential spreads to the neighboring region of the membrane, reinitiating the action potential. As depolarization occurs, the membrane repolarizes as potassium ions flow out. The depolarization-repolarization process is repeated in the next region of the membrane. Local currents of ions across the membrane cause the action potential to be passed along the axon.
So this is a diagram of the resting axon. Here it contains an Na plus channel and a K plus channel, which perform passive transport and flow by the voltage gradient and the concentration gradient. And here is an Na plus and K plus pump, which uses ATP to pump 3 Na plus ions out of the axon and 2 K plus ions into the axon.
Action potential depolarizes the plasma membrane of the synaptic terminal. After depolarization, the calcium 2 plus channel opens and triggers an influx of calcium 2 plus. An increased calcium concentration causes exocytosis of neurotransmitters. The neurotransmitters are released into the synaptic cleft and binds to the allosteric receptor of the ligand-gated ion channels in the postsynaptic membrane. These channels are opened and allow both the sodium ions in and the calcium ions out. Neurotransmitters are released and synaptic transmission ends when the neurotransmitter diffuses out of the synapse and is taken by the synaptic terminal by the enzyme.